have explained the cell cycle briefly, but of course, no cell cycle is meaningful without the cell death. The cell death is an indispensable part of our life. Actually, this is also the example I have given in the class. Think about a mouse, a human, and a blue whale. They all start life as a single embryo, single fertilized egg, and the size of this egg is not that different from each other. So this one becomes a mouse, this one becomes a human, and this one becomes a blue whale. The size of these organisms are they are very different from each other. Although they are all mammals, they have similar physiology and similar genetic and protein content. The size can be so different because they have difference in these three steps. Cell growth, not actually that different. Cell division, they have different cell division numbers, and cell death. So these are the part of the development. And by applying different developmental strategies, you can have different organisms. Okay? And you can create different body size. And we know that cell death is an indispensable part of development. For example, these cells in between the fingers, they have to die in the development, otherwise the organism would have a duck-like fingers. So for the sculpture, they have to die, although these cells are perfectly okay. I mean, they are healthy cells. There is nothing wrong with them. They are just in the wrong place. They have to die for a good sculpture of body. And in our body every day, millions of cells die, millions of cells are born. This is very normal. In the intestine, they are dividing, the intestine stem cells are dividing twice every day, they are dying every day. So this is how we keep our intestine healthy and fresh, which is normal. Or think about the tadpole. This tail has to be gone because although this tail is functional and healthy, it's gonna grow up, and when you're growing up, you have to change shape. This is normal that you lose part of your lifestyle. It's just time to give up. Okay. So this is the normal course of development, and this is happening all through the cell cycle, all through the development. So this kind of cell death is the controlled cell death and very clean way. So when these cells are dying, they don't create any chaos around them. They're dying in a neat way. But not all deaths are that way. This is, for example, necrosis. So when the cell is damaged, maybe it is burnt, or there's physical pressure, or there's a dangerous chemical or something, the cell just burst. You know, every cell is a chemical soup contained in a plasma membrane. You just damage it, so these nicely organized chemicals, they just burst out of the cell, and this is necrosis. This is a very dirty way of dying, and this is not preferable because these things, some of these enzymes are still active in the extracellular environment. They are going to catalyze reactions that is not nice, that shouldn't be there. The pH is going to change, the concentration is going to change in the extracellular environment. It is going to create inflammation. Again, it is not nice. So to prevent this chaos and inflammation, the cells die in a very clean way called apoptosis. Okay, check this apoptosis. By the way, Ebroja is going to tell this controlled cell death in a very detailed way. I will just give a brief introduction. Okay, it causes blebs. Unlike the cell is getting bigger and bursting in necrosis, it actually shrinks here. So you start with a large cell, it kind of gets smaller. All the organelles, they are fragmented and blebbed. This is a normal way it's doing. The surface is changing, the markers are changing. It causes blebs. You can recognize it under the microscope. Okay. By the way, it attracts the immune system cells. It gives a call, come eat me. not to create any stress around, to die in a dirty and to die in a clean and gentle way. It is asking the immune system cells, which are phagocytes, to eat it immediately so that it will be cleaned, and it's not going to create any stress for the multicellular organism. It is quite an altruistic behavior, I think. Okay, so one cell is giving up its life for the good of the body. The main molecule responsible of cell death is caspase. So caspase itself is a protease. And as you know, protease is an enzyme which cuts the proteins. And caspase starts its life as a procaspase. Okay? It has this site N terminus and this site C terminus. This N terminus site is cleaved off. Okay? And this is cleaved and changed shape. Only then procaspase becomes caspase. This is off. This is inactive, and this is on. So it is going under, not allosteric, this is going under post-translational modifications. Part of it is removed, part of it changes shape, and then it becomes a caspase molecule. So once the procaspase is activated into a caspase molecule, this is an active enzyme, and it is going to cleave other procaspases. This is a self-amplifying system. This is a positive feedback loop. Okay? These are called the initiator caspases. Okay. These initiator caspases, once they are active, they are going to activate other executioner caspases. Okay? Actually, every caspase created from procaspase itself is a protease molecule, and it's going to activate more and more caspase. And we know that many things are happening in the apoptosis, and each type of executioner caspase is responsible of one of them. For example, this kind of executioner caspase is maybe responsible of the cleavage of nuclear lamina, so that the nucleus disintegrates and makes the blebs. And the cytosolic proteins are also cleaved, so because they're not wanted to be active anymore, also the cell shrinks. It is done by another class of executioner caspases, and it is a self-amplifying molecule. So even if you have one single initiator caspase here, only one of them, it is going to create a very large number of executioner caspase. So this is why it is a positive feedback loop. Once it starts, it starts. The second thing is it is irreversible. Once you trigger it, once from the procaspase you make a caspase, there is no turning back here. It is going to start the apoptotic mechanism, and the cell is going to die. There is no turning back from here. Okay, every organism will die, and this cell will die most possibly very soon. Okay, previously I told you that every cell has a gun pointed to its head. It's more like a grenade. The pin is pulled, but here somebody is still holding it so that it doesn't explode. Okay, somebody is still holding it because every day somebody is telling it to still hold it, which is don't suicide. This message is called survival signal. Okay, the survival signal is it has to be given to every cell every day. The moment that the survival signal is not given to a cell, it is going to suicide. It's so ready to suicide. Every cell has a default suicide mechanism waiting to be activated. This is important because without this mechanism, we would be more prone to having tumors, cancer. This is a mechanism against cancer. This is a prevention mechanism. So How do we catch cancer? They somehow learn to escape this suicide signal. They somehow suppress this suicide mechanism. They synthesize their own survival signal, which is called, remember, autocrine signaling. They produce their own survival signal and they obey the survival signal, and then they go under mitosis. So normally there is a BUD and BCL2 molecule in the cell. When the survival signal comes in the form of AKT, remember the survival signal is taken from outside the cell in the form of hormones or paracrine molecules, local mediators, and then it is taken by the receptor tyrosine kinase, and the downstream effectors, including the AKT, is activated. Once the AKT is activated, the BUD is phosphorylated. BUD normally induces apoptosis, but once it is phosphorylated, it will not be able to induce apoptosis. And BCL2, it suppresses apoptosis and promotes the cell survival. So just by AKT, which is the downstream of the survival signal, it will say two things: don't die today. So inhibition of the apoptosis, and survive today, which is the signal to go on, you should leave. Okay, once the AKT signal doesn't come, this thing will be active. BCL2 is, you can be confused about BCL2. Many of them, they promote the cell survival, but some of them also induce apoptosis. Okay, there are different members of this family you see here, but there are different members, BUC and BUCs. So they can induce apoptosis, for example, in response to DNA damage. So what they do is that when there is DNA damage, and now you know that it is sensed by the p53, they are activating BUC and BUCs. With a large DNA damage, the cell has to die. And you know that in the mitochondria there is an electron transport chain and a cytochrome C molecule. The cytochrome C normally is working with the energy production. Normally it has nothing to do with the survival or cell death, but once the BUC and BUCs is active, they're going to make a channel here. Themselves, when they come together, they form this channel, and cytochrome C, which normally exists in the intermembrane space, it can escape from this channel to the cytosol. Okay? So from this original activity, which is just the carrying of electrons from the cytochrome C reductase complex to cytochrome C oxidase complex, now it has an entirely different characteristic. It escapes from the intermembrane space, it's like a sleeping agent, and go to the cytosol and initiate apoptosis. And how it is doing that, there are some adaptor proteins, the cytochrome C, which you see in the red, and the adaptor protein come together. And when they come together, they are not active themselves. 7 of them should come together. This is another measure against slow leakage. And this 7-armed apoptosome, the 7-armed killer, recruits to procaspase 9. There are different procaspases with different features, but they all work with the same principle. So once this procaspase joins here, it is activated, and this is called apoptosome. Apoptosome is a killing machine. Once apoptosome is here, the 7-armed killing machine is active. It is going to activate the executioner caspases. And these executioner caspases, they are going to induce apoptosis. Okay? And from this moment, the moment that this apoptosome forms, there is no way of turning back. The cell is going to suicide, definitely. Okay, of course, this is not the only way of inducing apoptosis. This is against DNA damage. And DNA damage is an internal information. This information is taken from inside the cell, but sometimes the order comes from outside. For example, here, they are healthy, they have no DNA damage, yet they have to suicide. It can also come from outside the cell, like the Fas ligand. So some cells have these Fas-dat receptors. They can be induced by the killer lymphocyte. The name suits, they are really killers. They have these Fas ligands. Whenever they attach a cell, whenever this ligand meets with this receptor, they are going to induce the procaspase 8. But that's the difference between these procaspases. Procaspase 9 is induced by the apoptosome, which is done by cytochrome C, escaped from mitochondria. And this is induced by the killer lymphocyte, which is an outsider. Okay, and this complex will be broken. The activated caspase 8 is going to activate the executioner caspases, and the cell is going to suicide. So this way, the killer lymphocyte can scan the tissue and tell the cells to suicide. If it doesn't like the cell, if it thinks that a cell is infected, or if the cell is out of control, or if there are too many cells in a tissue, it can tell the cell to suicide, and every cell obeys this order. They kill themselves, except the cancer cells. They are deaf, they don't hear this, so they don't suicide. All for suicide, we continue with